So yes, Hiroshi has thrown down the gauntlet. And yes, the hippocampus field is a little bit confused, but ultimately space is a fundamental organizing feature of our daily lives. And take, for example, every 80s adventure film hero, the first thing they say when they wake up in a strange place is, "Where am I?" So the ability to locate where we are in reference to the relatively unchanging outside world is important not only for navigation in the immediate present, but possibly also important for long-term episodic memories, so that we can place our memories of the who, the what, the when, within this context of the where. So today, I'm going to talk about the neural basis of spatial representation, and specifically the representation of location. And so I'll begin with a general tour of the cognitive map of space, as well as the spatial cell types in different species. And then I'll go into our work on place cells in the non-human primate in virtual navigation, and end with a teaser on some of the work that we're doing in place cells in real-world navigation. So the journey into the cognitive map of space begins in the hippocampus. And the very first cellular basis of the cognitive map was discovered in 1971 in the rat by the two Johns, O'Keefe and Ostrovsky. And so what they did was they implanted a rat with microelectrodes in the hippocampus, and they gave it a box to explore about this big. And what they observed is that there are cells that fire when the rat is in particular locations in the box. So it will look something like this if you overlay the activity of a cell onto an overhead view of the box. And blue represents low activity, and red represents high activity. And what you get here, this peak of activity, is called a place field. And different place cells have their own preferred spots, firing locations within the box. And together they cover the entire floor of the box. And each time the rat went into this location, the cell would fire reliably in the same location, suggesting that there is a memory for location, for space in general, within the hippocampus stored as a cognitive map. But in order to get from here to there, the rat needs to not only know where it is, it needs to know where it's headed, as well as how far it needs to go. And not surprisingly, there are cells within the hippocampal formation that code for these information. So there are head direction cells that fire in response to the direction that the animal is facing. So this particular cell, the aggregate activity of this cell tends to fire when the rat is looking at the southern wall of the box. And there are also grid cells, mainly in the entorhinal cortex, that fire in multiple locations within the environment that are regular and that tile the entire surface of the environment. And lastly, there are cells that fire to hard boundaries within the environment, and together these and there are also cells that represent conjunctive responses of place, direction, grid, and border information. And these really beautiful single-cell representations of a higher cognitive function have also been observed in mice as well as in bats. But we do know that the hippocampus is important in humans for spatial navigation as well. London taxi drivers who have memorized a very detailed spatial layout of the city have larger hippocampal volumes than those who did not pass the navigation exam. And famously, patient HM who lost his temporal lobe had severe impairments in his spatial memory. And again, patients of Alzheimer's disease also have severe spatial deficits. But when single cells are recorded in the human hippocampus, only a subset of the classical spatial cell types have been found. Grid cells have been found in the entorhinal cortex and the parahippocampus, but head direction cells and border cells have not yet been convincingly demonstrated in the human. Location-specific cells have been found, but as you can see, their responses are a lot fuzzier and much less specific than what you see in the rodent. And in addition to these 4 cell types, view cells that respond to, when the subject is looking at a specific part of the environment, have also been discovered. So there are important differences between human and rodent experiments that may account for these differences. So one important difference is that human subjects typically are patient populations who are undergoing treatment or investigation of epilepsy, and that may have unknown effects on hippocampal function. And a second important difference is that humans typically are put through virtual reality paradigms, much like this first-person driver task. And although many gamers will tell you that virtual reality or games, video games can be incredibly immersive, they still lack the vestibular stimulation that is important for head direction cells to fire. And a third reason could be that humans have much greater visual acuity than rodents, and also much more developed prefrontal cortices. So some people have suggested that humans may rely much more on visual rather than physical exploration of the environments. So these differences can be addressed using the non-human primate. So the primate brain is much more similar to humans, and we can combine techniques from both rodents and humans and use the precise single cell in vivo recordings to look at, together with virtual reality paradigms in the non-pathological brain. But a quick scan of the primate literature shows that place cells are similarly as hard to find in the primate as in humans. So in the ambulatory primate, only one study has shown these really beautiful specific place cell responses, but the majority of these responses don't seem to be stable over time. And a second group did not find any place cells, but instead they found spatial view cells that respond to views, particular views within the environment. So for example, this cell over here fires at multiple locations in the environment and at multiple orientations, but always to when it's looking at this section of the south wall of the environment. So, but on the other hand, Sylvia Worth and colleagues have found place-specific responses in the virtual reality paradigms in primates. So their animals explored virtual star maze starting from random arms, the ends of random arms, driving towards the center of the maze, and then having to locate a hidden reward. But you can see that the place cell responses, or the location-specific responses that they found, are similarly fuzzy. And this best example here fires when the animal is driving towards the middle, very near the middle of the maze. But a concurrent analysis of gaze information shows that this cell is also responsive to a landmark located about here. So he would typically see that when he's driving in from this arm. So this maze configuration makes it very difficult to separate gaze from location information. So given the lack in number and quality of place representations in the primates so far, we set out to ask 2 questions. One, are there place cells in the non-human primate, and can we separate them from spatial view responses? And second, are these responses different in virtual versus real-world paradigms? And this is a very preliminary result. So to start, we recorded hippocampal neurons in the macaque in a virtual reality paradigm, where he has to drive a point-of-view camera around the 4 pillars within a room, a square room. And there are, so this is a front view coming in from the south of the room, and this is an overhead view of the room. So there are 6 different posters which are target locations that he has to drive to, sprinkled throughout the environment. The locations are fixed, but they are, they are hung on the side of each pillar. So on any given trial, his task is to drive himself via the shortest possible path to a target location that we specify at the beginning of the trial. So it looks something like this. He's given a target, and he's driving himself now from the south end of the environment towards the donkey poster, which is behind the red pillar. And, oh, the sound is not working. But, hang on, let me see if I can get this to work. Can I get some help with the sound? Yeah. Yeah, it's on. Can I try again? Yeah. Okay, hang on.
Is there audio?
Yeah, there should be audio.
Giving your voice.
I'll try.
Setting kat mana?
Kau try cabut audio kau dan kau sama balik. Okay.
Yeah, if you have that, cool. Thank you. Okay, sorry about that. So again, this is, in this trial, oh no. Huh? Okay, thank you. Right, there we go. That signals the start of the trial, and he's moving himself to locate the donkey poster, which is behind the red pillar. And what you will hear as he moves between the red and the yellow pillar is the spike activity of the cell there. And overlaid on top of that, the green and red dots are the positions of his gaze within the environment, so what he's looking at. So behaviorally, it is clear that he has a good understanding of the task and also a good memory of the spatial layout of the environment. In blue, you can see that he has no trouble really completing the trials within the time limit, which is usually about 15 seconds. And then in red, you can see that almost always he's able to find the shortest path to each target location. And so we have a lot of data, but we've only analyzed a subset of it, and this is data from nine sessions across 9 days with about 400 trials each. And we've isolated single units from this data and computed spatial information scores for each of these cells and compared them to a 95th percentile shuffled spatial information that we got from 10,000 circularly shifted spike trains. And we have a total of 94 single units from the hippocampus, of which 26 are spatially selective. And that's a comparable number with the rodent literature. So, but on the other hand, the spatial information content ranges somewhere between 0.1 to 0.4 in the cells that we have. This is maybe a little bit lower than what we see in the rodent literature, but it may be because we have very few cells, we have more to analyze, or it may be because that there is something about virtual navigation that is different from real-world navigation. And so this is an example of 4 simultaneously recorded hippocampal cells. And the waveforms are typical of pyramidal neurons. And the 2 in the top row are examples of spatially selective cells. They have information content higher than threshold, and they also have very precise, tight firing fields. And the two in the bottom row are non-spatial, and they have typically high firing rates. So to give you an idea of the cell activity, we took the spike trains and overlaid them with the gaze information in this movie. So for this particular cell, you've heard it already. In this trial, it fires, when he moves towards the north, in between the yellow and red pillar. So that's one trial. And then in the next trial, he has to move from here to the pig poster, which is somewhere on the east arm of the maze. And again, it fires in between the red and the yellow pillars, very location specific. On the other hand, we have this non-spatial cell that pretty much fires everywhere in the maze without any specific preference. These are two of the same two trials, just to show you a contrast. And these are both cells recorded from the same electrode. So in order for any of this place memory to be useful in navigation, they need to be stable over time. So most of our cells, when we look at the activity split between the first half and the second half of the session, we find that they do correlate very well. Most of them have a correlation above 0.5. And while we haven't yet looked very rigorously at stability over days, here's an example of a cell. This on the same electrode recorded just one day apart with the same waveform and same field locations, suggesting that there may be cells that in our population that are stable over days. So then I just want to finish by talking a little bit about the next part of the project. So now that we have stable place representations in virtual reality, we can move on to see if they do fire the same way in the real world maze. So in order to compare this as closely as possible, we replicated the virtual maze environment. And instead of driving a first-person camera through the environment, the monkey now sits atop a robot platform that he can drive with a joystick. And here demonstrating this for us is our Primate colleague. And here are just 4 different views of the same movement. Here we have a phone camera recording of the setup. And here we have an overhead view of the environment. The red circle represents the target location. And in the bottom left, we have the gaze information that's captured by the eye tracker in front of the animal. And here we have a larger field of view that's captured by a GoPro camera above the animal. And at the same time, we record the neural information with the same microdrive assembly that we have in the virtual paradigm. And we also record the position of the robot throughout the maze with the LiDAR sensor. So with that, I will wrap up and thank Shicheng, my PI, as well as everybody who has contributed to both the collection of the data and the analysis. Thank you.
So for the interest of time, maybe one short question.
One short question. Okay. Hui Min, hi. Very nice talk and interesting to see your preliminary data. So I wanted to ask you, is there a conceptual reason to think that you might not find place cells other than just that in primates, because there's extensive literature in multiple other animals. So is there any conceptual reason? And if it is is there an alternative to, if not place cells, what kind?
Well, I'm not sure there is a conceptual reason why there wouldn't be, but I think many people have found that there are sort of more visual cell types than actual physical classical spatial cell types. So for example, I think Beth Buffalo's lab has found visual grid cells where they have the same regular tiling of activity, but restricted to a 2-dimensional visual field of view. So yeah, I guess now the work is to see if we can understand how the two relate with each other.
Okay, let's move on. Let's thank the speaker for excellent talk.